Next up, we have Dr. Olufemi Alabi to talk about watermelon crinkle leaf associated virus 1 and 2.
All right. Thank you very much. I'll start by saying that compared to the other viruses you've listened to, be it colitor viruses as well as the Napo viruses, watermelon crinkle leaf associated viruses are actually babies. They were just discovered in the last decade, within the last decade. And so there's a lot we don't know about their biology, especially. But what we do know, however, was, these were viruses that were picked up accidentally. So a group of scientists were conducting field surveys for cucurbit viruses in Henan province, China, in 2015 and 2016. During their 2016 survey, they encountered some watermelon plants, in that panel A, showing crinkling and mosaic symptoms. 2016, they also observed another set of watermelon plants showing a crinkling of the developing leaves. And compared to healthy looking plants, those plants actually have, this kind of bunchiness appearance to them, bunched up appearance to them, and the leaves were not fully expanded. So they did, what most of us do these days, taking advantage of the powerful tool called high-throughput sequencing that you've heard a lot about during this webinar series. And they isolated RNA and performed small RNA sequencing on these samples, both symptomatic samples from 2015 and 2016, as well as from LD-looking sample. And they found some viruses that are known to infect watermelon and other cucurbits. So that was no surprise there. But what was surprising was they found some additional sequences that did not match to known viruses infecting cucurbits. As a matter of fact, that did not match to most of the known viruses as at that time. They did a lot of, series of bioinformatic analysis, and eventually they came up with these two new viruses, that when they published our work in 2017, they named the virus found in the plant showing crinkling and mosaic symptom as watermelon crinkle leaf associated virus 1. And the 2016 sample showing just the crinkling and bunched up symptom, they named it as watermelon crinkle leaf associated virus 2. Those two viruses are related in some way. They belong to the same genus. However, they are so distinct that they represent different species. And of course, no virus was found in the LD-looking sample. But again, keep in mind, these two viruses were found in mixed infection in both cases. So they did a lot of, genome analysis, and they were able to determine that each of these viruses, whether it's crinkle leaf associated virus 1 and 2, their genome architecture are very similar. And it's basically composed of two RNA segments, one large RNA segment of about 6.7 kb, that is in the negative sense orientation, and the RNA2, which is much smaller, actually encodes two proteins, a movement protein and a nucleocapsid protein, and those have this, ambisense, strategy. There was no heavy, well, they did not pursue visualizing virus particles, but we can assume that based on their genus assignment into the genus Coguvirus, that these viruses also will have a filamentous flexus particle that have this particular dimension, if the virions were identified. So subsequently, it took about 4 years for another sighting of both viruses to be reported. And actually, this was a work that was being conducted by my then graduate student, Ms. Regina Hernandez. She was trying to document, viruses infecting cucurbits in Texas. The survey before our survey was done about 30 years before, and we kind of hypothesized that the landscape of viruses in Texas cucurbit crops must have changed. So we also employed high throughput sequencing in our survey work, And among the viruses that we found, including a lot of known viruses, we found this watermelon crinkle leaf virus 1 and 2. In most cases, a mixed infection, mostly in watermelon plants. So this represented the second report of both viruses outside of China and the very first record of both viruses in the United States. Subsequently, one thing I forgot to mention was, Regina designed some detection primers, which we included in our first report. And, we were very gratified to see that a lot of people subsequently used those primers to generate first reports of both viruses across different continents, multiple countries. United States, report of both viruses have been documented in Brazil as well. Of course, in China, in Spain, in Turkey, only virus 1 was reported. And in some cases, we have unconfirmed reports. For example, in Uganda, there's a recent publication that reported sequences, again, using high-throughput sequencing of sequences that resemble watermelon crinkle leaf virus, but they did not state which of those two viruses they found. So it is really very unclear whether it's WCrLaV-1 or 2 that was found in that particular Uganda report. But if that report turns out to be one of those two viruses, then that will represent the first African record. In the United States, the virus has been reported in different states. Both viruses have been reported in Arizona, Texas, Georgia, Florida. And in the case of Indiana, Oklahoma, one of both viruses was reported in recent publications. And you can see there, the citations for those first reports. So in terms of symptoms, this is where things become a little bit tricky. In the Texas situation, all cases of WCrLaV-1 and 2 that we have found have always been a mixed infection with other known viruses. And so we are unable to tease out very clearly symptoms due to either virus or both of them by themselves. However, studies done elsewhere, including from Georgia in the United States, have shown that singly infected plants, positive for, the watermelon crinkle leaf virus 1, may display symptoms that range from leaf crinkling to yellow mosaic patterns, to chlorosis, bunch it up, culling of leaves. The plants become stunted. Even the fruits may show some kind of deformation. But again, the thing to be careful here with is, a lot of symptoms may be influenced by mixed infection status of the plant or complications of some other abiotic stresses. So that needs to be taken into consideration. Here are just, some other close-up pictures. Again, these are pictures from Dr. Sudhir Bak in Georgia of single infection of WCrLaV-1 in both, fully expanded as well as young leaves. To your left is a symptom that was reported from Spain, again, positive for WCrLaV-1. And the zucchini plant you are seeing over there is based on a recent report from North Florida, again, of zucchini that was positive for WCrLaV-1, which actually represents the first report of the virus in that particular type of cucurbit. So a couple of things to keep in mind in terms of the epidemiology of these viruses is that watermelon is the primary host. At least most of the reports that we've received so far are basically emanating from watermelon plants. It doesn't mean that other cucurbits do not get it. For example, there has been reports of the WCrLaV-1 in in other cucurbits like melon, cucumber, as well as zucchini that I just mentioned in Florida. But primarily, majority of plants positive for both viruses are watermelon plants. Some of the work done by Regina also in Atis's work also showed that we can find positives in some wild cucurbits like smell melon, and even in some non-cucurbit weeds like pigweed. But again, we haven't confirmed the precise whether those hosts are playing any role as source of infection for the watermelon crop. Something to keep in mind also is the abundance of information that we are receiving from different parts of the world shows that WCrLaV-1 is more frequently detected than the type 2, and mixed infection of both viruses are very common, as well as mixed infections with other cucurbit viruses. The original paper from China, demonstrated mechanical transmission for WCrLaV-1 at a very low rate. Only one of about 40 plants, was positive, but they were able to demonstrate that the virus was able to move systemically within the plant that was mechanically inoculated, and the symptom was reproduced. There's no evidence of mechanical transmission of WCrLaV-2. There was also a recent report from Spain where they performed high-throughput sequencing on aphid samples taken from watermelon plants, and they were able to pick up WCrLaV-1, but we have to keep in mind that detecting the virus in an insect does not translate to that insect being implicated in transmission. And so more work still needs to be done to figure out the vector of these two viruses. A more recent work that was done in Brazil actually showed very conclusively that both viruses are seed transmitted. There's always been speculation that they are probably seed transmitted, but what the colleagues from Brazil have done is basically to do a very rigorous study where they were able to demonstrate seed transmission at a very high rate, especially for WCrLaV-1. Compared to WCrLaV-2, as you can see in that bar graph. And the other thing also to notice here is cultivar differences. So it seems to be that there are different cultivars respond differently in terms of their seed transmission rate. The virus was not just found in seeds as a contaminant on the surface, but was also found in different parts of the tissue. So this may actually explain one of the reasons why the virus seems to be very widespread within the short lifespan of its discovery. In terms of the diagnosis, we are basically the tool of trade for the diagnosing both viruses is basically based on molecular assays, particularly conventional RT-PCR. There are published primers, especially the primers from Regina's work has been extensively used and seems to work very well for most people who are investigating the distribution of these viruses. But again, always keep in mind that it's always good practice not just to score a plant based on amplification of the product of the expected size, but if this was a first investigation, to actually consider sequencing the fragment to confirm virus identity. And as you have probably realized in this talk, a lot of reports coming from different parts of the world is based on high throughput sequencing, especially for labs that are equipped to conduct, to use those, to use that particular assay. In terms of management, again, these are two recently discovered viruses. They are still evolving. There is a lot of things that we do not know about them. For example, there is a lot of question of whether they actually impact cucurbit production, either by themselves or in mixed infection with other viruses, whether they are contributing to symptoms severity. So there's a lot of things we still do not know about their direct impact. There's still a lot of things that we do not know about their biology, including the possibility of them being vectored, by an insect. There are anecdotal information from different parts of the world that there may be some possibility of field spread occurring. The original paper from China, suggested that trips may be involved. But again, these are all conjectures, so there's still a lot of work that needs to be done to investigate possible attribute vector. We do know, just like I've shown you recently, just in the last few slides, that it is both viruses are seed transmitted, which probably contributed to their rapid global spread. And so seed L testing will really be something that needs to be considered moving forward. And again, no information on genetic resistance in both, but the fact that different cultivars respond differently in terms of their seed transmission rate indicates that there may be some promise in looking for sources of resistance. So that's all I have. I would like to express my appreciation to students and postdocs in my lab who have contributed to some of the data presented from Texas. as well as to grower and industry cooperators for access to their fields, and my colleagues that have collaborated with me, and of course, the funding agencies that funded some of our work. And again, just like it's been shown before, we have actually a fact sheet on both viruses that was co-authored by myself and Dr. Sudit Bag. So you are welcome to please visit and learn more about these two emerging viruses. Thank you.
That was actually done before the seed transmission was reported. So if you download the PDF, that will not be in there, but we do hope to get the online HTML version posted, updated in that to include that information.